as technologies were becoming more and more advanced, it was no longer sufficient for each lab to have one microscope that they would apply to everything, when there are all these different types of microscope that are ideally suited to a specific biological question and specimen. It makes much more sense to have them all located within one central facility, and also to have people with expertise in microscopy so we can advise all of the researchers on what's the best kind of microscope for them to use for a particular project. The Bioimaging Resource Center was established as a center to house commercial systems. For a few years, I was actually trying to find some kind of light sheet microscope that would be useful for live cell imaging, but there was no one commercial system that we tested that we found would actually accommodate all of these different kinds of specimen. And so this is actually where the Beckman grant has been completely transformational.
We definitely want to encourage the additional developments that have been made possible through the Beckman grant, and we are taking advantage of those both in terms of optics and imaging and in terms of image analysis.
And so we chose, instead of buying one instrument that would only fit a certain number of people's needs, we chose instead to fund 2 people, new people to join my group.
My role was to design and build the optics lab with the help of plants operation at Rockefeller University, and build the prototype microscopes.
It became evident in the last few years, particularly with the arrival of latest microscopes, and people are acquiring so many images, that a course will benefit with a dedicated image analyst.
The Beckman Foundation Award allowed us to set up a new satellite laboratory, which is in newly allocated space.
This place was a basement, so we changed most of the things, the design, from electric circuits to curtains to ceiling tiles to Ethernet cables.
We don't want to just pay a lot of money to get a machine out of a box, but we would much rather be capable of either modifying existing machinery or building it de novo, as Bejat is now.
But then he'll be able to keep developing them and change them constantly according to the needs of a specific researcher, so that this instrument becomes a living, organic instrument, as it were. It's changing all the time, it's not static.
These prototype microscopes, they usually have custom parts. You need to fabricate those parts. So in order to do that, I needed some help from the machine shops. There are 2 types of parts that are needed. Some of them are bulky and some of them are small. They both need to be very accurate and delicate.
We didn't just need an optical engineer who was good at building systems himself. We needed somebody who was going to be able to communicate with the researchers as well.
There is a need for educating researchers about microscopy, how it works, because most of them come from background that has nothing to do with optics, lenses, cameras, lasers and things, but they are operating those systems.
So the SCAPE microscope was developed by Elizabeth Hillman at Columbia University. They're actually sweeping an oblique plane of light across the specimen very, very quickly, and the detection is sweeping across as well. So you don't have to move the specimen or the stage.
The only thing that is turning is this galvanometer here.
The specific thing we were doing is looking at the activity of nerve cells in the brain of the zebrafish. And the nice thing is, with that type of microscope, one can see a field of hundreds or more of neurons by hundreds, so 10,000 or even more neurons simultaneously.
And also we can modify that system in our core facility for the other groups at Rockefeller University who needs this type of modality for their imaging needs.
And so Jacopo is working on this really important question of how do insect repellents work.
I study specifically how these mosquitoes are repelled by commercially available repellents such as D-tempicardin, and I'm using the SCAPE system for looking at the neurons in the leg and how they respond to these different stimuli.
And again, this is a difficult preparation. We didn't have any microscopes in the lab that could see through that thick brown cuticle, the covering of the mosquito.
The neurons in the leg of a mosquito are organized in 2 layers, and therefore I need to look at both layers in a very fast fashion. And this microscope allows me to do volumetric imaging in a very fast way.
Originally the resolution was not high enough, so I had to modify some of the lenses and tweak the knobs and change the optics so that he can see the 2 rows separated.
The second microscope that we built, the lattice light sheet microscope, was designed by Eric Betzig and Wesley Legant and colleagues at Janelia Research Campus.
So instead of just beaming a light at a cell, there are multiple beams of light at different angles making a lattice that are all striking it at once.
We get a higher speed because the lattice creates a light sheet, and by moving the translation stage you can have fast volumetric imaging.
With the latest microscopes, people have been acquiring lots and lots of images, and now we are trying to quantify the images. So not only the observation, but also we want numbers out of them, and we want to do some kind of statistics to see whether there are some subtle differences.
The processing for this is very elaborate, it's computer intensive, and it takes true expertise.
Ved understands biology and microscopy and image analysis. This is really important for this particular role because he needs to interact with the researchers and understand their biological projects in order to be able to derive all of the necessary information from the images that will help them to interpret it.
And so VED has been absolutely key to many of our projects.
And we're currently working on a pipeline that allow me to look at where are the cells in the data that I collected and how do they behave, meaning how do they respond to the different stimuli over time.
In our case, working with VED has been really powerful because he's enabled us to tweak this algorithm in certain ways that I really don't think that we'd be able to do on our own.
So ClearMap is a computational pipeline for the analysis and registration of whole brain mouse images, which usually come from a light sheet microscope.
What ClearMap allows you to do is to say, this is a picture of the whole brain, which with each of the cells individually labeled. What ClearMap does is it aligns this to an atlas. It says, based on what we know of the brain, this spot that you detected corresponds to this brain region that we already know.
This pipeline is a bit complex for a typical biological researcher because it's written all in Python, and it works most efficiently on a Linux workstation.
And so you need both the appropriately equipped computer power, but then importantly you also need someone who is enough of a software engineer to be able to fiddle with things, to be able to adjust the algorithms in certain ways.
So these are SCMOS cameras, and when you use the sensor chip completely, you generate tons of data.
Where we used to collect hundreds of megabytes or maybe a few gigabytes of data in a session or day or week, now we can collect terabytes of data in a few hours.
So that's an enormous challenge to manage the data. So to tackle this problem, we implemented Omero.
Yeah, the Omero project is, a way of managing both the data but the metadata attached to images. So we can track, from the microscope what were the parameters used, but then beyond that, I think very importantly, how the image was actually analyzed, manipulated, going forward to the publication.
So users can not only store their data in Omero, but they can also use it to organize their data, view it, and share it with their colleagues or PI.
Really the role for me in this was to try and help establish the community side of this and the training side of this in Rockefeller University.
So as part of the outreach at Rockefeller, we organize different seminars and workshops, and I have been particularly involved in organizing image analysis user group meeting.
It's actually sparked an entire new community of imaging scientists at Rockefeller getting together to discuss these problems.
So yeah, I'd like to take the opportunity to really thank the Beckman Foundation for supporting this project.
We don't know what designs are going to be released in the future, and that's why it's so important to have a person and not an instrument. Who knows, in 2 years' time somebody might develop some amazing microscope that just transforms the way that we can image samples. And at that point, we'll then be able to implement this design in our own lab. So this allows us to stay really at the cutting edge of science. And it's been tremendous to see how that's actually been impacting the community here at Rockefeller and the kind of research that can be done here. And we're very excited to see all of the different people who are going to come in the next few years from Rockefeller and all around New York City and beyond and use these instruments.
Thank you, Beckman Foundation.